My name is María José Saiz Abajo, and in these slides I want to summarize the main results obtained in baseline project within the work package focused on plant products. Baseline proposal on sampling plans for plant products to address selected safety criteria. Detailed results and all the data can be found in the deliverables derived from this work package that can be downloaded from the official webpage of the project. First of all, I want to mention the main objectives of this work package. The main objective was to review current sampling plans for selected plant products, to identify risk factors and their performance objectives and food safety objectives in the selected plant products, and to assess the need of new or adapted analytical methods to ensure plant product safety. During the early stage of the project, the products, processes and hazards to study were selected. In the table we can see the final selection. 4 different products were chosen: lettuce and ready-to-eat lettuce, pistachio, cabbage and apple. The biological hazards studied were Listeria monocytogenes, Salmonella enterica, E. coli 15787 virus, and the chemical hazards selected were pesticides, mycotoxins and acrylamide. CNTA, National Center for Technology and Food Safety, is the leader of plant products tasks. It is a private non-profit center located in the north of Spain. Its aim is to contribute to the development and innovation of the agri-food industry, offering technical and scientific support to improve the quality and competitiveness. In the slide we can see some pictures of the laboratories and pilot plant where the baseline project has been carried out. At the beginning of the project, the review of legislation and sampling plans was performed among the EU countries participating in the project. In the map you can see a red dot over the countries participating in plant products work package and a black dot over the countries participating in baseline project but not in this work package. The main results obtained after the review is that there is no official surveillance for all selected risk product combination in the European countries. In general, viruses, E. coli 157 and acrylamide are not tested in the selected products. The official surveillance is performed according to European legislation or national legislation based in the European one. However, the sampling plans differ between countries or even between regions in the same country regarding frequency, number of samples and lots to be tested, location and sample size. In general, there are important logistic problems in the sampling of big lots, for example pistachio, that make the correct application of legislation very difficult. Some of the problems are big lots mixed during unload or afterwards in the silo or warehouse, so traceability is lost, partial unload of lots in different harbors, safety of workers, big lots unloaded during different days, etc. The uncertainty of sampling process is crucial to perform a correct sampling, and it is not always well known or considered. Few data were obtained from producers regarding number of samples and frequency of sampling. The main conclusions drawn in this stage were that there is a need for improvement and definition of the sampling procedures. The performance objectives and food safety objectives for some of the selected plant products has a combination. This is one of the main objectives of baseline project. The improvement of analytical methodologies together with a deeper knowledge of the real distribution of the different hazards for the selected products along the different chain steps have been the major outcome of the project. This will enable to plan effective sampling schemes with the correct number of samples to be collected in order to fulfill the required POs and FSOs. The dissemination of these results to authorities, regulators, producers, food business operators and consumers will provide information to establish more efficient safety policies, which will be translated into a higher consumer confidence in food safety. In order to derive performance objectives and food safety objectives for the selected plant products, different tasks were performed in order to provide experimental data and information needed to model the different hazards. Several experiments were performed to obtain experimental data to cover different data gaps that were not covered by existing data. We studied the transport of samples as a source of variability. We performed field trials and market studies to determine sampling uncertainty in pesticides. We studied the effect of some processing treatments on the studied risks. We performed different sampling plans on the same samples, looking for mycotoxins in pistachio, and we studied the obtained results. And with all this data, we calculated the hazard model distributions. From these distributions, we derived the corresponding POs, PCs, and food safety objectives for the different hazard product combinations. And finally, we proposed fit-for-purpose statistically based sampling plan for each case. It is important to take into account that we have translated into numerical and statistical data the real risk existing when a contaminated lot enters a production line. The dispersion of the contamination and the probability of being detected had been studied. Among the different experiments performed, we collect new data about cross-contamination. Two main studies were carried out, one on fresh-cut ready-to-eat lettuce and another one on fresh apples. For the first one, we tried 3 different levels of contamination. The samples were produced in a pilot plant simulating the industrial process, and the whole amount of samples of the process were analyzed. Distribution of the contamination on the final product was then calculated. For the apple study, we simulated a commercial process for fresh apples at a laboratory scale. 2 levels of contamination were tested, and water and sponges or drying material were also analyzed to determine which is the ideal sampling plan to early detect contamination in a processing line. In the study of transport of samples as a source of variability, different variables were considered: temperature, seasonal evaluation, transport container, and time passed from the purchase till the laboratory. And well, it can be concluded that temperature of refrigerated sample changes significantly when they are transported from the point of sale to home in a common plastic bag, usually used by consumers, whereas isothermal bags are more appropriate to avoid fluctuation in the temperature of refrigerated samples. Regarding pesticides, different field trials on apple and lettuce were carried out, and quantification of pesticides in unit crops and composite samples were analyzed. Market samples were also tested of cabbage, lettuce, and ready-to-eat lettuce. All these data were used in the project to determine sampling uncertainty and investigate the impact on food safety on the application of different sampling plans. On the other hand, studies on mitigation of pesticides in fields and with different processing procedures were also performed. 2 different crops were chosen and different processing such as washing, scalding, cooling were applied. It was demonstrated how these processes can decrease the final content of pesticides in the product. Regarding mycotoxins and pistachio, 2 different sampling schemes were tested to measure mycotoxins in pistachio. Official sampling plan according to Commission Regulation 178/2010, and an alternative sampling plan more reduced, usually performed by industries to control their raw materials. These sampling schemes were applied on 8 different lots, and the same lots were analyzed before and after roasting. The alternative proposed sampling plan led to significantly lower results when comparing aflatoxin B1 and total aflatoxin content, mean values of raw and roasted lots obtained from the two sampling plants, due to the reduced probability of taking a contaminated portion in the sampling. Just to introduce the information that will be explained next, the Commission Regulation 2073-2005 in Article 2C defines food safety criterion as the criterions defining the acceptability of a product or a batch of foodstuff applicable to products placed on the market. The food safety criterion must be able to accomplish one or more clearly defined objectives, such as to assess the safety of the food, the adherence to good hygienic practices and HACCP requirements, the acceptability of a food from external country when the history of the product is not known, and the compliance with the food safety objectives and/or the performance objectives. Based on these safety criteria, the different sampling schemes can be derived in order to fulfill the proposed PO and FSO. The different product hazard studies will be presented next. In the light of the experimental results obtained in the project and other from literature and the state-of-the-art of the general term safety criteria modeling approach was adopted to derive different safety criteria along the food chains of ready-to-eat leafy green vegetables. Therefore, a deterministic approach was selected to derive a potential food safety objective, which was used as benchmark to elucidate performance objective at specific process steps within the food chain of ready-to-eat leafy green vegetables. To this end, the following equation was applied, where cases corresponds to the number of reported cases associated to VTEC species in the 27 EU countries. P is the prevalence value for the pathogen. D is the ingested dose. N is the number of consumed servings, and R corresponds to the probability of illness per ingested cell taken from an exponential dose-response model with the form of the second equation, where P ill represents the probability of becoming ill after ingesting a certain amount of cells C. The value R is derived from the in vivo studies, epidemiological or in vitro studies data. Dose is defined as the product between concentration and serving size. Therefore, the equation can be rewritten as the third equation shows, where C and S stand for concentration and consumption and serving size respectively. To estimate food safety objective, the factor P by C was calculated as the function of the remaining parameters, that is, cases N, S, and R, which were calculated by using the total figures reported from the whole of the 27 EU countries. According to this approach described before, different food safety objectives were calculated for VTEC and Salmonella. Food safety objective of Listeria monocytogenes is already included in the EU regulations. These values are -5.8 log CFU per gram for E. coli 15787, -3.2 log CFU per gram for Salmonella, and 2 log CFU per gram for Listeria. In order to identify possible POs based on the proposed food safety objectives, simplified stochastic model describing the food chain for ready-to-eat leafy green vegetables was built. For that, a typical process of ready-to-eat vegetables was considered as it is described in the flow chart of the slide. In this work chart, it is included each of the processing steps that will be afterwards modeled. For each step or module, the variables used to construct the model were described based on data generated in baseline project and collected from scientific literature, which are included in Table 1. For example, for unprocessed product, the distribution considered was a normal distribution based on different scenarios considered. For Listeria monocytogenes and the step of washing, a model was considered to quantify the logarithmic reduction of CFU per gram from a study of Carrasco and al. Carried out by one of the partners of the project. The strategy followed to find the concentration levels in the food chains allowing to meet the proposed food safety objective consisted of simulating different fixed values for concentration at different steps along the food chain of ready-to-eat leafy green vegetables. These fixed values were plotted against the 95th percentile of the final concentration of the pathogen in the food at the time of consumption. The POs were determined by interpolation in the resulting curve. In the case of Salmonella and E. coli 157, the contamination levels at consumption were expressed as the product of prevalence and the 95th percentile of concentration, while for Listeria monocytogenes, as food safety objective was given in terms of concentration, the output was expressed as the 95th percentile of concentration. Following this strategy, 2 performance objectives and 1 performance criteria for the watching step were calculated. The same approach was applied for apple. In this table, you can see the final values calculated for performance objective, performance criteria derived from the food safety objective value for each hazard product combination. Results obtained for the different combinations of biological hazards and food suggest the great influence of the specific conditions at factory on determination of potential POs and PCs, especially the contamination levels at factory enabling to meet a specific food safety objective strongly depended on the contamination dispersion level in food and the efficacy of applied reduction steps, such as washing or disinfection. From that, the approach followed to derive POs and PCs was based on those values obtained at worst conditions of contamination, for example, high dispersion or prevalence, so providing higher POs and PCs values with the higher safety margin. The next step then was to derive sampling plans from the proposed performance objectives, performance criteria, and food safety objectives. The sampling plans were calculated for the different hazard product combinations. They were based on experimental data and they were calculated according to statistics. We have modeled the real risk on the different products, and statistics have been applied to compute the number of samples needed to detect a certain amount of contamination entering a processing line. In the case of fresh apples and Salmonella, from the POs and FSOs calculated, different sampling plans can be derived taking into account different scenarios possible. The results of the samples to be analyzed are shown in the table. The conclusion that was when the number of samples is very high, it is useful to analyze the sponge material or the washing water as good indicators of contamination. For ready-to-eat leafy green vegetables and Salmonella and E. coli, the portion log normal distribution was used to describe the contamination of lots with different mean concentration and standard deviations. In this case, and for the different scenarios, the number of samples ranged from 1 if the level of contamination is high and standard deviation of 0.5 or 1 to 100 of samples in the worst case. When the contamination is very low and levels of standard deviation of 0.5. The level of significance was 0.01 and the amount of sample to be analyzed of 25 grams. For general cases, 10 or 15 samples are enough to detect levels of contamination from -5 to -1 log CFU per gram at a standard deviation of 2. Finally, for listeria and leafy green vegetables, Poisson log normal distribution was applied according to preliminary tests carried out and collected data. The level of significance here was 0.05 and the amount of sample to be analyzed 25 grams. In the first plot we can see how the number of samples increased as the level of contamination became smaller. However, in spite of the low mean values, with the prevalence value derived from our studies, 18 samples were enough to reject a contaminated lot with a significance level of 0.05 at the lowest mean level, -6 log CFU per gram. Changing to the case of aflatoxin in pistachio, EU regulation established maximum amount of aflatoxins allowed in initial imported raw materials and the food safety objective after the roasting and salting process. The food chain includes different steps such as sorting or roasting that decrease the aflatoxin concentration. In particular, in the study carried out in baseline project, it was demonstrated with a wide sampling plan that roasting process or pre-roasting at 135 degrees and 20 minutes of roasting at 165 degrees decreased the total amount of mycotoxin in 75%. The main conclusion gained was that the sampling step was divided into sampling, subsampling, and subfractioning. Low variability was associated with the sampling step, probably because the size and number of elemental samples was enough. In fact, the last European regulation about methods of sampling and analysis for the official control mycotoxin levels in foodstuffs reduced the size of the aggregate sample from 30 to 20 kilograms. On the other hand, high percentage of variability was attributed to the subfractioning step, suggesting that either better grinding and mixing could be achieved, or more bigger subfractions should be selected to reduce this uncertainty. EU regulation 401/2006 established that a certain criterion of EU regulation 1881/2006 must be applied on results corrected by the recovery percentage and the analytical uncertainty, and referred always to the edible part of the nut. Sampling uncertainty is not considered in the final value and it should be considered on the PO determination. If sampling uncertainty is considered, to reach a food safety objective of 10 micrograms per kilograms of total aflatoxin content means to set a PO after processing less or equal to 1 microgram per kilogram. However, this is not the main inconvenience, because the problem can come from the values that could be established to the raw materials and at the entrance of the importing country. Are these values affordable? Finally, in the project we also have worked on the development and improvement of different analytical methods to detect and quantify some of the selected hazards. The improvements in analytical methods is directly related to sampling efficiency and the number of samples needed to detect an hazard that can be reduced. In particular, real-time PCR methods were developed for detection of different human pathogens present in plant products. Some of these methods have currently been validated in an interlaboratory European study. The multi-residue method for pesticides detection and its validation protocol for plant products have also been described, as well as the detection and quantification method for acrylamide in nuts. The whole protocols and the practical requirements necessary to trustfully apply a standard method in a laboratory have also been described in the corresponding deliverables. Finally, the main conclusions obtained in baseline project for plant products are the following ones. For biological risk, new molecular methods for detecting and quantifying human pathogens have been developed and intra-laboratory validated. Food safety objectives, performance objectives, and performance criteria have been proposed for different hazard product combinations. Different sampling plans have been derived from different scenarios based on the built models. All the data and models produced have been introduced in the baseline software developed. For chemical risk, we have studied a comparative of different sampling plans, official and industrial, in terms of uncertainty for mycotoxins in pistachio. We have performed the definition of the possible POs and FSOs for mycotoxins and pistachio. We have proved that there is a significant reduction of aflatoxins during roasting process. And we have studied pesticide field planning and sampling of different plant product groups, data regeneration for modeling and sampling schemes for developing. The following deliverables with all extended results mentioned in this short presentation can be downloaded from the web page of the project. If you have any question, you may also contact the Work Package Leader at CNTA, Maria Jose Saiz. I hope this information has been useful for you. Thank you very much.